This past week, we've been reading about enzymes and had learned from Chapter 8 of Campbell and Chapter 5 of Barron's AP Review that enzymes are important in regulating metabolic pathways, and without them, organisms wouldn't be able to grow, reproduce, organize, or maintain homeostasis. We began to wonder how certain factors affect the rate of enzymatic activity and if we could create an experiment to study this. The main purpose of us performing this lab was specifically to test the effect of concentration of enzyme on enzymatic activity rate. After we had created our lab experiment, we set up a null and alternative hypothesis. Our null hypothesis was if the concentration of the yeast changes, then the rate of the reaction won't change. Therefore, there will be no change in the volume of the balloon. Our alternative hypothesis was if the concentration of yeast in the solution increases, then the enzymatic rate of decomposition of starch into carbon dioxide will increase. Therefore, the volume of the balloon will increase. The independent variable in this lab is the concentration of the amylase. The dependent variable is the increase in volume of the balloon over a fixed amount of time. And the other dependent variable is the height of the bubbles. The control variables would be the temperature of the water, the amount of sweet potato puree used, the type of balloon used, the amount of water used, the type of yeast used, and the duration of each experiment. Our control group will be when the concentration of the yeast solution is 0 grams per milliliters, and the experimental groups will be changing only the mass of the yeast used in 2.5 gram increments.
So here are some images of procedures, materials, action plans, and visual setups that help us succeed in this experiment.
The purpose of this experiment is to test how certain external factors, in our case, concentration of yeast, affects the enzyme activity rate of amylase. After conducting the experiments, we found that as concentration of yeast increases, the volume change of the balloon in time also increases. In our experiment, the 10-gram yeast solution had a diameter of 7 centimeters and a volume of 205.3 centimeters cubed. On the other hand, the reaction with 5 grams of yeast only had a diameter of 3.5 centimeters and a volume of 51.3 centimeter cubed. Furthermore, we also found that the speed of the reaction is quite fast, with the bottle with the highest concentration having its balloon completely filled within 15 minutes. When putting this experiment on fermentation in historical context, we can see that it had a great impact on scientific breakthroughs regarding cellular chemistry and the role of the enzymes on those processes. At the end of the 19th century, Edward Buchner discovered that chemical reactions responsible for fermentation occurred inside yeast, thus awarded with the Chemistry Nobel Prize in 1907. By experimenting with yeast and fermentation, the significance of Buchner's discovery can be truly appreciated. Our conclusion from this experiment is that the rate of decomposition of starch into gaseous carbon dioxide increases in a more concentrated yeast solution. Also, as the concentration of the yeast increases, the height of the bubbling of the reaction also increases. So let's analyze the trend of this experiment to see how the concentration of yeast affects the enzyme activity rate of amylase. For the null hypothesis, we propose that the concentration of yeast won't affect the enzyme activity rate. For the alternative hypothesis, we propose that as the concentration of yeast increases, the enzyme activity rate will also increase. In our experiment, looking at graphs for both original and repeated, the 10-gram yeast solution had a balloon with a volume of around 200 centimeter cubed. However, the reaction with 5 grams of yeast only had a volume of 51.3 centimeter cubed. Thus, we can conclude that as the concentration of yeast in the solution increases, the rate of the decomposition of starch into carbon dioxide will also increase. This conclusion can be supported by a figure regarding the process of fermentation inside yeast, in which glucose is taken into the yeast cell with the product being ethanol and carbon dioxide. Therefore, we can reason that with higher concentration of yeast, glucose in the sweet potato puree can be decomposed faster, resulting in a higher rate of reaction.
One future extension of this experiment is to explore the effect of the different amounts of sweet potato puree on the rate of volume change of the balloon while the amount of yeast used is kept constant. In doing so, we can explore whether the amount of substrate will drastically affect the rate of the chemical reaction that generates carbon dioxide. The second extension can be to explore the effect of the different temperature of water on the rate of the volume change of the balloon while keeping other variables unchanged. The yeast solutions can be activated using water of differing temperatures. After a fixed amount of time, the volume of each balloon can be measured to calculate its rate of change. The third extension could be using different liquids with varying pH to activate the yeast. The purpose of changing the pH is to examine how external factors other than concentration will affect the rate of the reaction between yeast and sweet potato. Different liquids of varying pH will have different effects on the amylase enzyme. The differing pH of the liquids might cause a conformational change in the shape of enzyme, which will prevent the reaction from occurring through denaturation, but it may also accelerate the rate of the reaction depending on the pH, which causes an increase in the balloon volume. The first error is the calculation of the volume of the balloon, which affects the accuracy of how much the concentration of yeast actually affects the rate of reaction measured by the production of carbon dioxide gas. The second error is the inconsistency of water temperature, which might cause a slow activation energy of the yeast during the experiment. And the third error is the inconsistency of the amount of water and sweet potato used across the different bottles in the experimental groups because the amount of reactant is different throughout the bottles.